Hello, welcome to the Science is Everywhere Festival. Together with my colleagues from Rock EDU, Noggin, Miss Steph Science Club, Pueblo, and BioBus have put together an amazing experience for you. This is our Egg-Mosis event in the series. If you miss any event in the series, go to www.biobus.org/sciencefestival to see them on YouTube. I am Miss Steph from Triple F Empowerment Inc. And today we will do Egg-Mosis. I have with me today Daniel, and I have Latasha here with me today to answer your questions in the chat. And guess what? You can ask questions in Spanish. Daniel, can you say hi?
Hola. Hi, everyone.
Chat. And guess what? You can ask questions. Fabulous. In Spanish. And Latasha, can you say hi?
Hola.
Hi.
Yay, I'm so excited to have both of you with me today. And of course, you all online. And like I said, I'm Miss Steph, and I'm more than a little bit silly, so don't get distracted by the shenanigans, okay? We're going to have a great time. And today I'm going to talk to you about osmosis and the human brain. Can you say osmosis? I know you can. Say it. If you don't say it, I'm gonna know. Osmosis, okay? And like I said, the human brain. And so we're going to have an awesome time today with this activity. And as you can imagine, I called it Egg-Mosis because it's the word osmosis and egg together. The egg is going to be our brain model. Can you imagine that? The egg as a model for the human brain. And I'm going to talk to you a little bit about how that is. Okay, so first let's make sure we know what osmosis is, because some of you might be thinking to yourself, I don't know what that is, but you're brilliant and you can learn a little something today. And I have this very, very simple diagram to represent osmosis, okay? Osmosis is the movement of water from what we call from hypotonic to hypertonic. And hypo just means there's a lot of molecules, okay? A lot of soluble molecules like salt or water or sugar. And hypo, right? You see there's very few dots, okay? Meaning there's less, okay? Less sugar or salt or other water soluble molecules that are inside of the solution, okay? And this is a picture, this very fabulous purple line is a picture of a membrane. And guess what? Our brains, okay, have a membrane just like eggs do. And so, like I said, today we're going to use the egg as a model of the human brain. Now, how is an egg a model of the human brain? Well, I just talked to you about a membrane, okay? And that's a very thin layer that's under this protective shell. Imagine that the shell of the egg is like our skull, okay? And as you can see, I have my egg carton. My egg carton is another protective layer. It protects the shell, right? And so our skulls are the equivalent of the eggshell. Now, can anyone put in the chat? I know that you're out there and I know you're active, you're with us. Can anyone put in the chat the apparatus, the device that we might use to protect our skulls? Hint, it's something that you should wear every time you ride a bicycle, go rollerblading, go skateboarding. It's something that we should wear. Does Does anyone know what that is? What that is? Yes, that's a helmet. Is that what you put in the chat? It's a helmet, y'all. Okay. So our egg carton is like the helmet, and the eggshell is like the skull. Okay. And like I said, there's this thin layer called a membrane under the eggshell. And for us in our human anatomy, it's the blood brain barrier. And that's the thing that lets special important molecules go across it. And it keeps other things out. And that works through osmosis. Remember, high concentration to low concentration. Now, let's take a beat and think about what I might need to do to this egg to get to show you the action of osmosis and how important it is and how exciting it is. I think I might need to do a science activity. What about you? I think we should do it. Okay. Now, while I get my materials out, okay, can someone say why we need to be protective of eggs and our brains? Can you put that in the chat? You're here with us and we are excited that you're here. So please feel welcome to put things in the chat as far as why you think we have to be protective of our brains. While I get out my materials. Okay. So thank you for sharing your thoughts in the chat. Great job. All right. I have my materials here. So let's talk about them a little bit. I have my labeled containers. Cups. I know, fancy word for cups. I have eggs. I have several eggs here. Okay. I have vinegar. I've got corn syrup. Okay. How viscous that is. I wonder if that might mean that there's a lot of a really high concentration of molecules inside of here. Of my sugar molecules. And then I have some water. Water. It's going to be my hypotonic. So this is my hypertonic liquid, and this is my hypotonic liquid. And you'll see why that's important later. The first thing that we're going to do, though, is we have to change this egg. We have to change this egg, right? We have to get rid of the hard shell protective layer. And notice how I'm being very careful with my egg. Okay, I don't want it to break during my experiment, or rather my activity, right? During my activity. Okay, so I'm going to put some of my things back to the side. You'll see them again later. So as I said, I have this labeled. The first thing I need to do is remove this layer. Now, something that's exciting and so interesting about eggshells that's similar to our skulls is that eggshells contain calcium. Okay, and it's one of the minerals and things that helps the eggshell stay hard. And vinegar reacts with calcium and it helps break it down. So I'm going to keep everything inside still safe while breaking down the eggshell, I'm going to gently put my egg inside of this container. Now, for those of you who would like to try this at home, I want to remind you 2, make sure that you have adult permission and probably some adult help to make sure that you have permission to use these materials from wherever they might be in your home, okay? And to also make sure that you are making safe choices while we're pouring liquids and so forth, right? So again, that's the first thing I'm doing. I have phase 1 and I have vinegar written on my container. So I have my egg inside of this liquid. Do you see the bubbles? What? The bubbles let us know that the reaction is taking place. Oh man, it's so cool. That's our first phase. I'm gently removing our hard, protective, calcium-based shell from the egg because I want to expose that special membrane. I'm going to put this to the side. Something important about this for my friends wanting to do this at home is that this step takes a long time. This step is going to take 24 hours. What? Yes, I said it, 24 hours. Okay. And some of you might notice vinegar has such a strong smell, and maybe you don't want your apartment or your home or wherever you're at to smell like vinegar. Here's a trick. I put my samples inside of the refrigerator overnight, and that stopped the vinegar smell from getting all over my apartment. Okay? So I'm going to put this down. All right? Vinegar and my egg. Gently, quietly. While I do this, Latasha, are there any questions in the chat? Anything I should know about before I move on to phase 2?
There's no questions, but we were talking about that the brain controls everything, and that's why we need to put helmets on. And also, so we were talking about the brain controls everything, and that's why we need to put helmets on. And also, they were worried about the brain getting broken. That's why we need helmets as well.
It's perfect. That's excellent, right? I think that's such a great observation and such an important piece of knowledge to have, right? When you have knowledge, you are empowered, okay? You get to make choices that are responsible. All right, and so I am so appreciative of how you all are participating in the chat with us. All right, now I have several things over here to help me do what I'm doing, okay? And so that's why I keep looking over here, just so you know. Now, that was phase 1, remember? We needed to get to break down that hard, strong, calcium-based shell. And we did a chemical reaction, right? We started a chemical reaction to break down the calcium and the hard shell. And now, after letting it sit for 24 hours or more, I now have an exposed egg. Okay. This is what the inside of the egg looks like when it still has the membrane around it. I'm going to be very gentle with this. Okay. The membrane. Okay. The membrane. Okay. Still there. Okay. Now, I'm gently, just like I did before, because we still, like we said, we are using the egg as our brain model. And you all so effectively put into the chat that we need to be protective of the brain. The brain has control over all of our bodily functions, right? And so if I'm treating the egg like it's a brain, I need to be gentle. And I'm placing it into now phase 2. Phase 2. All right. And remember, we're learning about osmosis in the brain. And now that our membrane, that thin layer that can let some things pass through it, has been exposed, now I can add my hypertonic fluid solution, right? And that's for this activity, today I'm using corn syrup. You can try other liquids, right? Other liquids that have a high concentration of a solid. And I'll show you in a little bit some other options that you might want to try. But today I'm going to do this with corn syrup. So I'm pouring the corn syrup, right? You see how I have my labeled container. And you might even notice that I've used this container before, but I washed it and I'm reusing it now as a responsible human on the planet. I don't want to use too much plastic if I don't have to. So I'm reusing this container and I'm adding my syrup. Okay. And so now this egg, my brain model, with its exposed membrane, is going to sit in this liquid, our hypertonic situation. Okay. And I wonder what will happen. And I wonder what this will look like in, yes, you guessed it, 24 hours. I know. I'm making these things sit for a long time, but yes, in 24 hours, I'm going to let this sit for 24 hours. I'm going to gently put it to the side.
Miss Steph, we have a question in the chat.
I love questions.
Um,
Scientists have questions, right, Latasha?
Definitely have
So
questions.
they're clearly a scientist.
I know. She wants to know, did the egg change color?
Excellent question. Okay. So we will answer that question together. All right. By me showing you an egg. Okay. That question is perfect for the next part of the activity. Okay. I'm going to show you the eggs throughout each stage because I've saved them. All right. So you see how this particular egg is a brown egg, and we know that eggs actually come in different colors. Usually in the store, they're either brown or white, and they're all the same on the inside, okay? And so this is what it looked like before. The membrane, okay? The membrane, let me show you this one, okay? So the membrane is kind of translucent. So it has some color to it, but you can kind of see through it a little bit, okay? And so you notice how the egg itself did not change color, but we exposed or revealed or showed you other layers of the egg. Again, that question is awesome and great and really thoughtful and reflective, okay? But no, the egg has not changed color. For this particular thing, I'll show you something later where the egg changes color. But the egg does not change color. We are just revealing or showing other layers in the egg. Okay. Any other questions, Latasha, before I move on to the next part?
There's not a question, but there's someone that noticed that the egg doesn't sink in the corn syrup. And we were talking about, they just made that observation that that was pretty weird.
The egg is less dense than the corn syrup. Okay. The egg is less dense than the corn syrup. And so things that are less dense will float on top of a high density substance. Ooh, fancy word. It's just a way of saying stuff. Okay. Yeah. So the egg was floating. So now, actually, that's a perfect, again, you all are asking these perfect questions that help us go to the next steps. Okay. I'll show you some of my samples here, because remember, I'm putting the eggs in different liquids and letting them sit. And so I have an array of samples here to show you of what happened over time. Yes? So let's do that part. Let's start with, and you'll get to see the various densities and all that type of stuff. Okay. I have my plates and I have them out here and I have them labeled. Okay, as a scientist, I have to make sure that I have all of my samples labeled so that I know what everything is. Okay, so we just looked at this egg, okay, and you notice that the egg, after sitting in the vinegar for 24 hours, the egg is about the same density as the vinegar. Okay. So let's take that egg out and put it here. Remember, we're learning a lot of different things. We're learning about osmosis and the blood-brain barrier and all those things, okay? And so I'm going to make sure that you get a chance to observe the impact, the effect, okay, of our hypertonic solution. So after 24 hours, okay, this is another sample. You see I have the date and everything. This is another sample. So this has been more than 24 hours, but after about 24 hours, it starts to look like this. So all of the water has moved outside of the egg, okay, across the membrane, okay? So the water has moved outside of the egg into the syrup. So this is a syrup sample that has had time to sit. And you notice I'm being very gentle. I'm going to take it out and set it on the plate so that you can have a better look at it. And you notice that it was still floating. Okay. And so the egg was still less dense than my liquid. Okay. Let's put that there to the side. Now, what would happen if I put one of these eggs, a vinegar egg, with the membrane exposed, into water? You might have that question. Well, guess what? I have an answer. You notice how it sank. It's at the bottom. So this egg is at the bottom. I wonder if there are any other differences if the movement of water across a membrane has made the egg look has any other differences in the egg. Hmm. Hmm. Looks like this egg is bigger. So the water has actually moved into the egg. Do you see it? It's very exciting. And of course, the egg is round. It's rolling. But this egg, you notice this egg doesn't roll anymore. Huh? It looks almost like it's deflated, like a balloon, like it's a deflated balloon, but it's not. All of the water that was in the egg through the process of osmosis has moved out into our hypertonic solution, which was our corn syrup. I love it. I get so excited. Now, here's something else I'm going to show you. Getting a little tripped up here. Such is life, it happens. Okay. I have this sample. I thought to myself, I wonder what would happen if I took a corn syrup egg after it sat in the vinegar for 24 hours. And then I put that egg and put it inside of corn syrup for 24 hours. And then I took something that looked like this and now put it in a hypotonic solution, a solution that has water in it. That's mostly water. And then I put a little food coloring in it. Why? Because color makes life better. Okay. So I wondered what would happen before I take this out. Can we get some predictions going? Does anyone have a hypothesis as to what will happen, whether or not I'm going to take an egg out that will look this size? Will it look like this? Will it revert all the way back to its original state? Will it look like that? What do you think will happen? I think we should give you all some time to formulate a hypothesis, a prediction. I think so. What do you think this egg will look like? Remember, it was a corn syrup egg. Okay, so it looked like this. And then I put it in some food coloring and water. I put it in a hypotonic solution. I'll show you again. I want to give you time to think it through before I reveal the egg. Latasha, do we have anything in the chat? Does anyone have a hypothesis that they're sharing?
Not yet, but I think people are just excited to see what was going to happen.
See it?
Yeah.
Are you excited, Latasha?
I'm excited. Food coloring is the best.
Yes, it's the best. It's one of my, is it one of your favorite science materials?
It is one of my favorite science materials.
It's one of my favorite.
Yes.
This is one of my favorite. And I actually used black for this. Okay, so let's reveal. Remember, I'm still being very gentle.
Good stuff. We got something in the chat. All right.
I love it. Yes.
I think it will drink the water back, so it will be normal or bigger. That was really Tina Tiny Tina.
Yes, thank you, Tiny
Ashley
Tina.
thinks it will explode.
Ooh.
Tashay thinks if the membrane is intact, then it will get bigger. There's someone who thinks it will be big and black.
Aha. All
So
right.
lots.
Very thoroughly stated hypotheses. First of all, I want to bring that to your attention. You notice how several of you added the vocabulary words, right? Membrane, okay? Membrane intact. Some of you were specific about not only do you think it would have expanded and absorb the water, but that it would also absorb the color. Well, let's find out. Again, I'm being gentle because this is an egg and we are treating our egg like a model of the human brain. Okay, so I'm going to hold the egg up. There we go. Excellent, excellent. So there we are. Okay. Our egg did absorb the color. Now, I want you to notice how the egg absorbed the color at the bottom the most, the food coloring, because I left it undisturbed in the refrigerator. Again, if you're concerned about how the smell might affect your home, which I'm a little smell sensitive myself. And so I kept my samples in the refrigerator. Okay, that way I didn't have to worry about any microbes growing, and I also did not have to worry about it smelling like all of these chemicals in my house. Okay, all of these various chemicals. So you see what happened. Okay, the color was absorbed into the egg. Okay, through the process of osmosis, the color and the liquid and the water. Okay, and so the egg has actually gone back to close to regular size and actually really close to the size of the egg that was in the water. All right, so I have 4 different samples, but let's bring out our regular egg for comparison. Okay, we're going to compare the egg in its original state. Okay, because as scientists, it's very important to do your activity, to put things through your process, right? Through the chemical reactions and so forth that we had happen. And then we are using this as a control. We're using this, our egg in its original state, the way I bought it from the store, to compare to the various steps and stages. Okay, that's what I'm going to do now. And then, of course, we will absolutely take questions. We'll take more questions. So excited that you are joining me and my colleagues for the Science is Everywhere Festival. We have several other activities that are available for you, and we'll talk a little bit about that later. All right. So this is my regular egg compared to the vinegar egg. So you notice that the hard shell is gone and the egg is actually bigger, right? Because there's water inside of vinegar. Okay, so the egg was able to absorb some of the water. All right. Next, we're comparing our regular egg to the corn syrup sample. So after being in the vinegar, then I put it in corn syrup And we had someone that made an awesome observation about the fact that the egg floats in the corn syrup. It actually stays on top of the corn syrup. And throughout my process, I actually was not pushing it down, but I was stirring it a little bit to pour corn syrup on top, okay, while it was, again, in the refrigerator overnight, okay? Then let's look at a comparison of I put the vinegar egg inside of water. And you see it's even more plump. It's even more plump than the vinegar egg was, okay? And finally, I thought it would be fun to see if I could restore, okay, this egg that looks like it's a deflated balloon to its regular size or bigger. And I also thought it would be fun to put in some food coloring and see what it would look like. And this is what we got. And we had some fabulous predictions concerning that. Lots of people put in their hypothesis in the chat, and they said that that's what they thought would happen. Okay, so your hypotheses were correct. You did a great job. So I think it's a good time to take some questions before we do a recap of all the things that I did.
Really Tiny Tina wants to know what will happen when you hold the egg for a month or a year. I think if you put it in the different solutions for a month or a year, what will happen?
So really Tiny Tina, you're doing a great job of thinking like a scientist. That's an awesome question. That is something that we could test, right? As a scientist, you can test it. You think to yourself, I wonder what would happen. You start with that question. You then make a hypothesis about it and then put it inside of those liquids and hold them and let them sit, okay? So that's how you would test it, your process. I have not done that exact activity. So if you have parent or family member permission, okay, you could try it. I wouldn't suggest going longer than a month, but if you put it in the refrigerator, okay, right, you can manage the smell in your home, okay. But you could put it in the refrigerator in a cup, like I used these cups, okay, and I put it in the back, and you can see what happens. These samples, just to give you an idea how long I've had some of the samples. I love that question. I love all questions. I'm a science teacher, so I like questions. I am going to put that one out. That one I haven't talked to y'all about. The samples I'm going to show you, I labeled them, okay? And so some of these samples have actually been sitting for almost a week. This is from the 13th, the corn syrup one. But the sample that the water was in, this is actually from the 9th, okay? So this egg, the egg that was in vinegar and then was in the water, has been sitting for the week, okay, because today's the 16th. And yeah, both of these eggs are actually over a week old, okay? Because I wanted them to sit for a very long time, so you'd be able to really see what happened with the process, okay? And then this egg, same situation, because it's gone through I had it go through several phases, right? It went from, let's review what it went from. It went from plain egg to being in vinegar and then to being in corn syrup, and then being in food coloring and water. So this sample was in food coloring and water since, let's find out, since I'm a responsible scientist, I labeled when I put it in. It went to the food coloring water, went to stage 3 on the 13th. Today's the 16th, right? So I did that on Saturday. Okay. So you can see, Tiny Tina, how they hit kind of a, trying to think of how to say this the best. They hit in like an equilibrium. Okay. So where after sitting, you might be thinking, what's an equilibrium? Or maybe you know that word, okay? But after sitting long enough for the reaction to happen, which is in the first 24 to 48 hours, okay, they then stop reacting. Okay, it's the best way that I can say that and describe that. We have Latasha, do you have any thing to add to that or more questions?
All right. Someone hypothesized that if you put it in water for a year, it will get big, 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 big.
I love that hypothesis, right? So this egg was in water for about a week. Okay, right? Not about a week, a full week. This is the one that was in the water for a week. And so the egg can get but so big, right? It can expand, but then there's a certain place to where it stops expanding simply just because of how not just big, but how much water the egg can absorb, okay, through the process of osmosis. Like there's but so much water that will go back and forth. Because again, that goes back to what I was saying to Tiny Tina about equilibrium, right? After a while, osmosis stops because it's equal on both sides. Osmosis only works when it's not equal on one of the sides. Okay, and once it becomes equal, it stops. Okay, and so the egg does expand to a certain capacity, and then it will stop expanding. Okay. Other questions or comments? We're having a great discussion. So excited to be with you all today.
We don't have any questions or comments right now, but I think I'm excited to see what would happen if you put it in like a rainbow of colors. What would happen? Would it be the same?
Right. I mean, that would be another extension, right? So let's talk a little bit about things that you could do to extend this activity. So first of all, in the chat, a lot of your questions are extensions in themselves. Extensions meaning what could you do beyond what I have displayed here, right? Because you're scientists. We're all scientists on this call. That means that you can think of a question of what would happen, and then you make a prediction, and then you test it and make your observations. If you wanted to, you could always come back and share your observations with us. Okay, so I think we should set up a little something. Okay. Uh-oh.
Sorry, Miss Steph, we have one more question.
Oh, I love questions.
How else does our body use osmosis?
Ooh, great question. I actually might need you to answer that with me. I'm going to start. So all of our cells have that capacity, right? And so we use water. I mean, osmosis is based off of of moving water or solutes back and forth. And solutes meaning the molecule, the different molecules, the things that we need to use in our cells. Okay. And so that's happening throughout our whole body, I would say, is what I would say. Okay. And so it's not just in the brain, the brain, it's happening throughout the cells in my whole body. Things are moving back and forth all day, every day. Okay. Is there a way that you would comment on that or expand on that, Latasha?
I think that's really great, and we can think about like when you stand in the shower or the bathtub too long, and your fingers get pruny, and that's another way that we can think about it. Just uh, just practically, you can see it on yourself.
Love it, love it, love it.
Yeah.
And there are other ways to see it in other organisms, right? Like if we put other organisms or other, especially like with plants, if we're putting plants in some water. But there's slightly different kind of action happening there, right? That's more capillary action. But yeah, the pruny fingers, I love that. I love that visualization, Latasha. That's fabulous. I'm going to use that.
All right.
Do we have other questions?
And someone was saying absorbing nutrients in your GI tract uses osmosis. And yes, so okay. What happens if you put, oh, I guess this is about the egg. What happens if you put it in soda?
In soda. So, couple of things that are likely to happen. First of all, if you were to put the egg, this type of egg, right? The egg that still has the hard shell in the soda, you are going to get some reaction. Because if we recall, the egg and our skulls have calcium in them. And so the egg has a really high concentration of calcium as far as that outer hard shell. So you would see some bubbling happen because soda is acidic. Then after it's sitting for some over after that 24 hours, right? 24 to 48 hours where the egg, that eggshell would break down, then you would start to get one of these types of reactions. And actually, truth be told, I would say you would get a bit of a combination of all of them. Soda, depending on whether or not you're using a soda that has pigment, right? So if you're using cola, you would get something kind of closer to this egg, right? Because the egg is going to expand a bit. It will absorb, because soda, even though it has sugar and other solutes in it, it's pigmented and it has water. Not enough water for it to be nutritious for us, but it does soda does have water in it. So you'll get a bit of that happening back and forth. If this, I mean, we're getting into some other things, but if the soda was left out long enough to where the water had a chance to evaporate off, it might become syrupy enough to where you'd get something not exactly like your corn syrup egg, okay, because the corn syrup was just straight up corn syrup, but you might get this egg with pigment and then kind of deflate a little bit. Okay, that question is fabulous.
It'll be a great experiment for them to do at home. And
I think there would be a perfect extension, right? So that's the thing that we want you to think about or consider, options that we're giving you. What can you think? Okay, what did you learn today? Feel free to put that in the chat or talk about it with a family member, okay? Or maybe turn and talk if you're watching from a class at school, okay? And what does this make you want to do or try? We've already talked about several things. Latasha was telling us how she's curious what would happen if we put it in different colors, right? Or put it in like somehow some type of rainbow situation, right? Um, we just had a question about soda. Maybe you could try several different sodas. Okay. Maybe an orange, a grape, a clear soda, and a brown, like a cola, right? What would that be like if you compare all four of those samples? And of course, you need a control. So maybe like a water if you put, okay. Um, what would happen if I tried it in salt? Yeah, right? I have salt, so it should have behaved like a hypertonic uh, solution. Okay. However, there was so much water in here that it still actually behaved more like when I put it in the hypotonic, right? You see how big that egg is? And all my salt sank to the bottom. Most of my salt is on the bottom. Okay. So you could try salts and other substances. And just another gentle reminder um, definitely, if you're trying this at home, ask for adult permission, okay? And ask them if they want to do it with you, right? Because wouldn't that be fun if you could talk about what you both learned, okay? And before we do anything else, let's talk a little bit about cleanup. Scientists must clean up. Okay, no one is going to come and clean up for me. So as a responsible scientist, I'm going to dispose of everything properly inside of my trash, okay? Um, I'm not going to eat these, okay? All right. So because of all the chemicals, yes, the chemicals we used were edible, okay? But in high concentrations, um, not so much. And also the egg, you notice I never cooked the egg. All right. So these are not things that are edible. These are things that we have learned from and had a great time seeing the results of. So before we do um, any other review, let's check and find out if there are any more questions in the chat, because you all have been very active and brilliant scientists today. I want to make sure that you get a chance to ask all of your questions.
So, is it just eggs that we can use? I wonder if we could use other parts from living things. Like, could we use plants in different liquids to see how they react or see if they react the same?
Yes. Actually, plants have some different, plant cells have different structures than our cells. And so you could actually do a comparison, okay, between like say how the egg behaves and how long it takes, because the egg takes a longer time to react and change than some plants might. So for example, going to this food coloring and water thing, comparing how the egg looks to maybe a piece of celery, okay, a piece of celery, or some other um, plants, a flower, okay, seeing if the flower, what part of the flower changes color, if any. Eh. Okay, so you could definitely do this with plants and various plants, right? Um, and compare how long it takes for it to react to the water or the other liquids. Other questions before we get ready to close out? All right. So Thank you so much for attending our Science is Everywhere Festival. If you enjoyed the experience, which I think it was an experience, I enjoyed it. If you enjoyed the experience, make sure you push the like button and subscribe, okay? Tell us how you feel about science in the chat. How did this activity make you feel? We plan to continue this festival next year, and we want to hear from you what you would like to see. Please keep in touch with us at sciencefestival@biobus.org. Also, check out our events throughout the year. All of our contact information is in the description of this video. Don't forget to tune in to our other events this week, and we'll see you next time.